Farm for Profit Podcast. Take a listen, have a blast. Farm for Profit Podcast. Learn about farming while having a laugh. Farm for Profit Podcast.
And listeners, welcome back to the Farm for Profit Podcast. This is Tanner.
And Corey's here.
In studio.
Yeah.
It feels darker today.
In the studio?
In the studio.
Did you could slide your lights up?
I could slide the lights out. It's one of the nicer days outside the studio.
It is. I can actually feel the warmth from the sun actually coming in. It's a nice week this week.
It is. It really is. Listeners, we hope you're experiencing some good weather yourselves. We thank you so much for following us, for connecting with us and trusting us as one of the resources to help your farm achieve higher levels of profitability, especially here in 2025. So if you do us a favor and you do find the content valuable, please share it with somebody. Send them a text message and say, hey, check this out. Or I didn't know this. And see if we can bait somebody else into listening.
Yeah, absolutely.
Leave us reviews. Of course, thumbs ups. We really appreciate those as well. If you've got topics, farmforprofitllc@gmail.com. But we've been told we got to keep these intros a little bit shorter.
Yep. Get right to the meat.
I want to know, Corey, do you know anything about what we're talking about today?
Yeah, and today we're talking about soybean cyst nematode, but I know that it takes a lot of yield away from my soybeans.
Yeah, right here at the top of my page, it says yield robber.
Yeah.
Unmasking the number one soybean yield robber.
So I've farmed all my life, and then I went to college at Iowa State University from 2006 to 2010. And that's probably when I started to learn about it in my internships and through my schooling there. And as I got out, I spent most of my normal career before I came back farming full-time in the seed industry.
Normal career?
Yeah. The podcasting is probably not a normal career path. And maybe I should say professional career, my short professional career. That's when I really learned about it and how much it affects yield. And then one year we had sudden death syndrome, and I've heard that that's can cause that. And when you have 40 bushel soybeans and you're expecting 55, 60 plus, it's not a good deal.
Yeah, I would say so. So let's introduce our experts that are going to help fill us in a little bit more and give our audience the information that they need. We've got Dylan here and Horatio. Welcome to the podcast, gentlemen.
Thank you.
Oh, thank you.
Since neither one of us asked you how to say your last names, we just went with first names only in the intro. So, Dylan, why don't you start off and tell us who you are and let our audience know where you're from?
Yeah, thanks for having me. My name is Dylan Mangel, and I'm the extension soybean pathologist at the University of Nebraska. And I'm actually from Nebraska, too. So I've grown up around row crop operations, and now I get to focus just on the diseases that we see on those soybeans.
Okay. Awesome. Thanks for joining us. And Horatio, yourself?
Yeah, thank you for having me. My name is Horacio Lopez Nicora. I'm the soybean pathologist and nematologist here at the Ohio State.
Yes, we were out at the Naked Conference last week, and we interviewed several people, and it's always the Ohio State University. So, yes, we get it. You're important.
It's branded, you know.
It is.
Where have you guys been?
We just got back from the Custom Harvesters event where operators from across the country couldn't stop talking about Steinbauer performance modules.
Yeah, they're seeing up to 25% more power, better fuel efficiency, all with smarter injector duration and not altering the rail pressure for safe and reliable performance.
If you're looking to maximize your machine's potential, go to steinbauer.us and tell them Farm for Profit sent you to get yours today. Appreciate you guys coming on the podcast and hanging out with us. And I didn't necessarily know that this is a topic we needed to talk about. But as I dove deeper into it, we need to talk about it. So, Dylan, why don't you give us the background? What is the overview of soybean cyst nematode?
Yeah, so soybean cyst nematode, it's a non-native pest. It's a non-native parasite to the U.S., and as soybean production moved across this country and really, really ramped up, this pest sort of moved with it. It's a nematode. It's basically survives in the soil, and it's going to parasitize those roots, just sort of sapping yield away from those nematodes. And the big reason we need to talk about it is because it's oftentimes going to steal some level, often significant levels of yield without noticeable above ground symptoms. So it's not going to turn the plant red or purple. You can't really just see it's out there. You might just be missing some yield, and you might not know why. And sometimes it might be low enough that you don't really even think about it, but it's still taking that money out of your pocket.
What's the history of it? So you said it's not native. How did it get here? If it just comes.
Yeah. So the history of how it got here, I think there's probably differing ideas on how it got here, but it sort of showed up in the Carolinas. It showed up the East Coast, and then it kind of followed the Missouri River up and Mississippi River up, actually the Mississippi mostly, and spread out from there. In Nebraska, for example, where I am, we didn't have it till 1986. And it hit one of those edge counties along the river. It followed that river up, and then it's consistently worked west from there. And now we pretty much have it in every county that's growing soybeans in the state. We're still adding some new ones as soybeans continue to move west, but it's going to move around with soil. It's going to move, even if wind is blowing soil, it's going to move around. So it's able to consistently march along, and it's ultimately just going to follow everywhere beans go.
So Horatio, how's it do that? He mentioned wind. Does it attach itself to soil particles, or how's it move like that?
So yeah, like Dylan mentioned, there's several hypotheses of how it's moving so fast, because the fact that it's moving, it makes sense. We move soil around, it's gonna move around. So equipment that we don't clean very well, it's gonna move the nematode around. It sticks to the soil, and anything that can move that will move the nematode. But how did it move so quickly, right? How did it expand throughout North America so quickly? And a different hypothesis is that it can also travel through that digestive system of these migratory birds. These may be picking, roots and particles of soil that may contain those cysts, which are the dead females that contain the eggs, and these will survive through that digestive tract, and they will be dispersed. That's another hypothesis, right?
Mm-hmm.
But one thing that we were talking at the beginning is that, this is an animal. It like us. It's an animal, and it penetrates the root system and overtakes the plant machinery. If we go and make thin cuts from those roots where the nematode is feeding, because it feeds from particular cells that it modifies, so it turns those cells into feeding sites. And if we make very thin sections and look at under the microscopes, those cells are completely modified, resembling transfer cells. These are not cells that we are supposed to find in the root system of any plant. They're cells that we're supposed to find in the embryo, in the seed. So the nematode is really taking control of the plant machinery and making it believe that in my root system, that's where you need to feed your progeny. Of course, I'm making this extremely more simple than what it is, but right. We don't see symptoms like Dylan say. We have beautiful soybean plants losing 30% yield, and we just thought we had the best season of our life. And we left, on average, $300 an acre below ground?
So is the yield loss coming from damage of cells or turning the cells into doing something that they're not supposed to be doing?
That's a great question. We have a physical damage, which is a penetration of the nematode. It has a stylet that we'll use to break into the cells and the root system, migrate to the vascular cylinder of the plant, where you have the phloem, where you have the xylem. So movement of water and nutrients are going up and photosynthesis are coming down. And the nematode sets these feeding sites very close to that region. So double punch there, right? But more of the damage is caused by these modified feeding sites. and it's way more intricate than just saying, you know, it modifies a cell, right? There's evidence of research of, you know, taking control of hormonal system, right? So some of the major hormones like auxins will be up and down regulated. So it's fascinating, right?
It's almost, isn't that like the definition of a virus that changes cells and does that, isn't it?
It's a parasite. It
is a parasite.
does a great
That's
job.
what it is.
Yeah.
That's what I was looking for. It's a parasite. So it's a nematode parasite, kind of.
Right.
Yes, it's a plant parasitic nematode. So that's how we're gonna call it normally. So, and not only that, we can add obligate parasite. It requires a living root to survive and to reproduce. You take the nematode out of that root that is feeding on, and it will not survive. You try to grow it on something that is not alive, you will not be able to grow it. Very differently than with other pathogens of soybeans that we deal with, right? You mentioned SDS, for example, is caused by a fungus. That fungus can be easily grown in a media that is not alive. It's still a parasite. It can parasite the plant, but still you can grow it on a media that is not alive. Soybean cyst nematode is an obligate parasite.
Does the damage to the root allow for that SDS fungus to get into the root easier?
That's a great question. The mechanism of the interaction is something that we are still trying to better understand. So the reports that we have and the evidence of interaction are from very indirect observation. What does that mean? Well, when we have them both together, we see more SDS symptoms showing up. Or SDS symptoms, which are those interveinal chlorosis, yellowing in between the veins of the leaf, show up earlier when we have a lot of nematodes. Understanding how that really happens, which is a mechanism, is still something that we're trying to figure out.
Mm-hmm. So Dylan, it only survives on soybeans, though. We don't have nematodes in corn or wheat, or do we?
There's a lot of nematodes, right?
But not this particular type?
Yeah.
Yep. So we have different nematodes in those crops for sure. But this one, it really specializes on soybean. But I will say there's likely exceptions where it's finding weedy hosts or other similar plants to soybean that it's also able to grow on as well. So think about that with weed control as well. Just imagine the difference in, corn and wheat are a pretty similar monocrop. Soybeans are quite different when it comes to how related they are. So those nematodes aren't crossing over necessarily. But if you've got other similar crops or other similar hosts out there to soybean, they might be growing on those as well.
So let's talk through some of the ways we can control this, or maybe the history of controlling. I know there's been several things through seed traits or genetics and that kind of stuff. Who can talk about that the best?
I'll talk about it.
Okay.
So soybean cyst nematode, it's been around, it's spread, and it actually started to become quite an issue in the 1990s. At that time, research, science, industry found a resistance source that was able to control this. So that first step of control was introducing a single resistance gene into a lot of the varieties that were out there. They put it in a few at first. Slowly, they dumped it into more and more. And eventually, it sort of ended up with that one resistance gene in all the varieties, or in most of the varieties. That resistance source was called PI 88788. So if you look in a seed catalog for soybean, you'll see a column. It might say SCN, and it'll say PI 88788 in a lot of those varieties. That's still the case today. And that worked really well for quite a while. The problem came in the early 2010s, and it's the same problem we see all the time when we rely on one thing to control a wild population. Those nematodes evolved to overcome that and reproduce on that resistance source. So PI 88788 degraded in its effectiveness, And we really, at that point, it was a turning point for our organization, which is the SCN Coalition. And how do we combat that degradation? What do we do next? So at that point, we looked around and realized there's a bit of a gap. Resistance is the most economical way to manage a disease, varietal resistance. And we really didn't have anything available. There were a couple other resistance genes available in the germplasm. So we really needed to put this group together to sort of build a stopgap for how can we identify what's coming next? How can we get resources to growers? How can we get them to start, maybe trying some of these new resources and preventing that yield loss on their farms? So that's how our organization started. I guess I should mention what the SCN Coalition is. We're basically a public-private partnership that was basically formed just to build awareness of this. This is a situation, academia doesn't always have common goals with industry. We have different ideas at times, but this was a situation where growers didn't know they had this pathogen or this parasite. And from extension, we want them to know it's there so they can make those decisions to manage it properly. Industry had the same goal. So it was a time when we overlapped. So we built this group with funding from soybean checkoffs, state, regional, national, and then we also work with some industry groups as well to just build awareness and get as many growers aware of this disease as possible. So that's what our group does.
So you're still working on the next solution?
I think a lot of people are working on that next solution. And it's going to come in different ways. So if we just look at the general way to manage soybean cyst nematode, we think about that varietal resistance first. The next thing is rotation. Rotating to a non-host crop can make a huge difference, even if it's just 1 year out of soybean. Now, I know there's not a lot of soybean on soybean growers out there, but a normal corn soybean rotation actually can go quite well. Just a single year is going to knock those numbers back by a lot. The next thing is considering a nematode protectant seed treatment. In that gap where we started to see resistance fall off, industry came in quickly and it started to fill that in with a lot of new products that are going to be basically nematicides. We're also seeing new generations of bionematicides, biologicals targeting these. So there's a lot of new stuff. Considering one of those, and then the big follow-up for management is just testing to see if you have that nematode still. You can't see it unless you have very extreme cases. You might see stunting in your fields, but like Horacio said, you could get up to that 30% yield loss, which is that's a lot, right? But without those noticeable above ground symptoms. So testing is really important to see if this population is managed properly, if it's staying under control, or if you need to take more extreme actions with management.
So I'm still interested in the trait that was, or the gene that was entered, the PI88788, and now it's the new one I think that I've seen is like peaking. Is that we talk about?
Correct.
How does that deter? Does the nematode not like the taste? Does it kill it? How does that work?
Yeah, that's a great question. And so the nematode will penetrate the root. So sometimes we think, that resistance will avoid the nematode penetrating the roots, and that's not the case. So you put a tomato root and the soybean cyst nematode will go inside the root. It will penetrate. It will use the stylet, which is like a needle that it has on the head, to break those cells and migrate to the vascular cylinder. When the nematode penetrates a susceptible soybean and does this migration to the vascular cylinder to establish this feeding site, it will probe cells with that stylet. It will release effectors, right, to engage in that interaction of host pathogen. When that happens, the nematode becomes sedentary and cannot move anymore. And that's how it's going to complete its life cycle. The susceptible soybean will feed the nematode for its entire life. Now, at one point, the males will regain this worm-like condition, will get out of the root, and will fertilize females. The males won't feed anymore. At least we don't have evidence that they feed. they will fertilize the female. This is a sexual obligate parasite, just like we are. We have to have sex to reproduce. Soybean cyst nematode have to have sex to reproduce. Now, this is very important because every sexual recombination means that there is a possibility to shift virulence, to adapt to new traits, to become more resilient. So this is great from the perspective of the pathogen that can adapt very well. Now, what happens when we have a resistant soybean? The nematode will penetrate the root of a resistant soybean, will migrate to the vascular cylinder, will start probing those cells and engage in this interaction with this plant. As a matter of fact, the nematode will become sedentary because the nematode thought that it actually established that feeding site. Now, this resistant plant, once that happens and the nematode cannot move anymore, kills all the cells around the head of the nematode, which are supposed to feed the nematode, starving the nematode to death. I mean, this is very cruel, right? I don't know if these are time to talk about this event, right? I don't know who's listening this show, but it's a very cruel situation, starving the nematode to death. We don't care, right? But that's how resistance works. That's how resistance works. Now, the nematode can adapt, like we mentioned, and overcome that. And it happens just exactly how Dylan was mentioning, the overuse of the same source of resistance. I mean, PI88788, which has amongst the many genes one locus that is the ARCHAG1B that confers a lot of resistance to the nematode, has been used for over 30 years. I mean, just think about telling someone, go and spray this same active ingredient to manage your wheat for 30 years. We are going to exert a selection pressure, and that's what we did. The population of the nematode that we were able to very effectively manage with PI88788 is not there anymore. The virulence profile shift to a different type. And that's the type that now prevails across the soybean producing region of North America.
That's wild.
So we're losing efficiency with PI88788. We identify other sources of resistance. So when we talk about PI88788, PI stands for plant introductions. These are horrible soybean plants that have traits that we like. We don't like to grow them. They grow horribly, very viney. As a matter of fact, you can have it in the house for like an ornament. I actually have it at home as a decorative plant, but horrible. We cannot grow it, but it has a trait that is resistant to soybean cyst nematode. We can use it to introgress it in high-yielding soybeans that we like to grow. Peking is another one. And there are 7 that we know of that have resistance to soybean cyst nematode, right? Now, Pekin is an alternative like we just mentioned, but we have evidence that if we overuse that, like we did with PI88788, we will lose that effectiveness very, very soon, even more so than PI88788.
So you said there's 7 of them, but I've only seen 2 in my seed selection. So where's the other 5?
You know what? That's a great question, right? And these 7s are the ones identified as we screen for these germplasm. We just go and we have access to these germplasms of weird soybeans, and we test them against soybean cyst nematode, and then we can identify. Now, important thing is that they have to have different mode of action, if you want to put it in a very simplistic way. That the genes associated with the resistance have to be different, and that's the case for all of these. And they appear in chronological order. Peking was the first one to be identified, PI88788, the second one, all the way to cloud. Now the question is, I'm only seeing 2. What's happening with the other one? In most cases, they're not easily introgressed in high-yielding, high-producing soybean. In other cases, some of the populations of soybean cyst nematode we have already developed the ability to reproduce on these sources. And therefore, the search for new sources of resistance, for understanding better mechanisms of resistance, and maybe combining it with other tools.
Mm-hmm.
Now, you did mention as well the seed treatments. We should probably talk about that. I'm guessing like Elevo and like Saltro and things of that nature. Are you talking about those, or are there others?
So there's others coming online. So industries, that's sort of a rapid response to this problem. Those two products came out. There's definitely new ones coming out. Timerium from Syngenta is something that's going to be coming out, and I think we've seen that in trials in the field in our research. And they're being pretty confident in that. I think we'll see that in the next couple of years. I'm not sure of the exact timeline. There's also bionematicides that we've been looking at as well. So these same companies, some additional ones, they're putting those products out there. When it comes to these, I think everyone's seen biologicals pick up in the last couple of years. It's what we've seen with them so far is maybe some level of effectiveness, but I think when it comes to biologicals, you've got to have lower expectations as well. So those products are often marketed as pairing with other products to get a more complete control. But as time goes on, and as we realize the limitations of resistance in the short term, industry has filled that gap with these kind of products.
So same question as how the peaking works. You said it in nematicides. Is that killing the nematode?
Yeah, so I think about them, just like an insecticide. In fact, I often think about nematodes as insects. They're basically small worms. So if you can poison them, if you can kill them, it's going to work like that. Now, a lot of these products have different modes of action. And, in fact, it's just the same way with fungicides. We have quite a few mode of actions, a lot of different chemistries. But ultimately, the individual products are there to kill those nematodes.
Okay.
So the most precise term that I would recommend, to encompass all these products would be seed protectant, because some are not strictly nematicides, right? By definition, nematicides is a product that will target that animal and kill it. In many cases, some of these active ingredients are fungicides in their origin, right? So fluopyram is an excellent fungicide that, in vitro has activity on egg hatching or the nematode itself, right? But then when we think about this integrated management and we think about our field having SCN, which is a big problem, that doesn't get rid of all the other pathogens that we also have in that field. And just enhancing that root health comes very handy when we use a seed protectant, right? So we may be, like you mentioned, Elevo, for example, it's great for SDS, sudden death syndrome. And we know that there's an interaction that may be going on between the nematode and that fungus. So that could, give some extra protection there for the plant.
Is there certain soil types or regions that these things thrive in, or are they just pretty much anywhere where you grow soybeans?
Yeah, they've spread pretty much everywhere rapidly. They definitely have preferences. I think nematodes in general, they need some sand in that soil so they can get around. They need pockets in there. So compared to a bacteria, these are much larger. So they need to be able to move around or swim around in those soil pockets. So we typically see worse damage in those sandy soils.
What about life cycle? I mean, you said moving around. How far is this little microscopic worm moving in its lifetime, and how long does it live?
So, yeah, go ahead.
No, go ahead, Horacio.
No, so the life cycle in general, it's 1 month. I mean, this is rough, right? Depending on the conditions you give it, it's going to be 27 days. It's going to be a little bit over a month. But in 1 month, you will have the female completing that life cycle, being fertilized and laying eggs. On average, 250 eggs. That's a field average. And that can happen 3 to 4, maybe 5 times in one season, which is, huge in the sense that we start with very low soybean cyst nematode. And that's a comment that I always get from my growers when we have conversations about this. I don't have to worry about soybean cyst nematode. I have very little soybean cyst nematode. And when you have low numbers of soybean cyst nematode, for some reason, that relationship with the ability to reproduce very fast is inverse. So it's inversely related to the reproduction factor, which means the lower nematodes you have, the faster they will reproduce. In one season, that number can explode very easily. And it makes sense, right? You have more root for fewer nematodes, and each one can complete a life cycle in 1 month and lay on average 250 eggs.
How long can an egg?
We talk about
how long can an egg stay there before hatching?
That's a great question. You know what? There's a gap in research in terms of survival of the eggs, but not only survival of the eggs, also the hatching mechanism, which is fascinating. The nematode, those eggs within the dead female, when the female dies, it will enclose eggs within that female. And the dead female is what we call the cyst. It melanizes, it oxidizes, it becomes very protective of those eggs. And those can last for a long period of time. We're talking about 10 years or more.
Wow.
Now, the hatching process is one that we, I think, still have more to learn. We know that there is a batch, a cohort of those eggs in the soil that will hatch when the temperature, hits a little bit warmer, point. So, you're getting to spring, coming out of winter, it's getting warmer. Those eggs will hatch. There's another cohort that will only hatch if it smells soybean, if there is exudates of the host. And there's a third cohort, which is an enigma, which will hatch whenever they want. It's a time dependent. You provide warm temperature, they don't hatch. You provide soybean exudate, they don't hatch. They hatch whenever they want to hatch.
Jeez.
And if you think about it, that's a great mechanism for survival, right? Because what if we want to put a rook exudate that is extremely resistant? It will pass and it will stay in the field. So once you have soybean cyst nematode, in my opinion, you're hitting an action threshold.
Farmers, you do it all. So should your technology. Level up your equipment with the latest innovations from Ag Leader.
From sure force downforce control and sure speed high accuracy planting to right spot individual nozzle control, Ag Leader has you covered.
And now, with the new In Command Go displays, you get unparalleled visibility and control, keeping critical information at your fingertips. Upgrade today and reap the benefits this season.
Visit agleader.com or contact your local Ag Leader dealer to get started.
What was it, the cicadas or whatever that only hatch every 12 years or 19 years or whatever it is?
Yeah.
It's just on a rhythm, on a cycle. That makes it hard to combat if there's various reasons as to why they hatch.
Right.
So you kind of identified, I think, the answer to my next question. We're going to walk our fields. We're going to pull some plants up. Is that the only way to identify we have it, is once we see the cysts on the roots, or how do we know we've got it in our field?
I'd say just pulling plants isn't even enough, because depending on soil type, especially unless you have really sandy soils, those cysts are going to fall off really easily. So it's actually pretty difficult to dig those. And, we'll try it sometimes. We'll dig them up and we will very carefully pull soil off and see if we can see those. But even then, that can be deceiving if you pull that up and have nothing there. Really, soil testing is the only consistent option to see if that's in there. Fortunately, a lot of states have free or checkoff funded soil testing programs for soybean cyst nematode. I know we have one here in Nebraska. Any Nebraska farmer can bring our lab soil samples and we'll tell them if they have it. Horacio, you guys are the same in Ohio, correct?
Yeah, correct. In Ohio, we provide up to 2 samples free of charge for growers. They can submit their samples and we'll process those for soybean cyst nematode, and then we'll report numbers of eggs per 100 cc of soil, cubic centimeters of soil.
And I'm not sure off the top of my head, but several other states also have programs. So if you have questions, I think the first place I'd start is with your local extension in your state or your local extension service, and they could probably guide you right to the most economical testing option in your state.
I mean, I'm pretty sure we had to pay for my soil samples that we had and we sent to Iowa State, but it was very economical. It was less than $30 a sample. And when you really start to think about how much we spend on soil samples for nutrients and other things like that, and you spread that across all your acres, it's very, very cheap to know, just so you can start get a game plan, right? That's what we want to do.
Yeah, and that's a great comment, Corey, because we have been trained as growers to pull samples for our soil lab nutrients, right? We do it routinely, right? That same soil samples, we can pull a subsample and send it to a nematology lab. We collect it in the same way. That task that really going out and getting the soil sample is the most time consuming part of doing it. Once we have that, we can split that and send it to the soil lab for nutrient analysis and to the nematology lab to test for soybean cyst nematode.
So let's go through the sampling process. When is the best time of year to get samples?
Well, the great thing about this nematode as far as sampling is it's always going to stay in the top of the soil profile. Some of the corn nematodes will move up and down at different times of the year, so you got to really time those perfectly. This one, you can sample any time of the year and at any point in your rotation. But I'll suggest that the point at which those populations will be at their highest is towards the end or at the end of a soybean season. So if you're just starting out and you want to check, grab those soil samples in the fall right after you harvest. And then you can see what they are at their peak level. But if you do grab them off cycle and you want to follow up in 2 or 3 or 5 years and see if they're moving still, grab them at that same point in your rotation, and then you'll have a point to compare them with.
Yeah. So I know on mine, I was sampling ahead of soybean planting, fields that I planned to go to soybeans. So it was mostly corn, coming out of corn, because that's when I'm going to be growing them, right?
Right.
So
that's good, and you could do that too. But it's really good to know when we read those results that these are coming out of corn, because we know those numbers have fallen a lot. So we can read into those numbers differently based on where you are at in that rotation.
Yeah, I saw you grab your pen because we didn't tell you that before the conversation started. So let's jump into Corey's.
I'm pretty sure that's what the rules said to do. Was because actually we were going to go sample our other co-host who's not here, David's field, and he's been corn on corn, and he was planning to grow corn again, and it said plan to sample ahead of planting soybeans. I said, oh, we're not going to go to your field because you're not even growing soybeans.
Yeah.
So.
But we were curious, right? The introduction to this topic was interesting to us. So that's what we wanted, is we wanted some actual data that you guys could kind of talk through with us. So I know, at least Dylan, you've seen the numbers for 6 samples that we pulled from Corey. Can you run through those, or do you want Corey to kind of run down that list?
Well, if Corey has any background information on those fields, maybe it'd be good for him to share some of that.
Yep. So I sampled 6 fields. And the reason I sampled them was because it was kind of a cross-section of our farm. Like my trout field, it had a pretty long history of corn on corn. It was corn on corn for probably 9 or 10 years until like 2018. And then we've been in a kind of a normal rotation since then. And then Let's see, Home and Harmon and Bill's 80 and Chad's were kind of our normal rotation, I would say. So I just kind of wanted to get a cross section of like, what does it look in our corn soybean 50-50 rotation? And then this Crum one, it was a brand new field to me, and my landlord purchased it, and it's kind of 30 acres. It's kind of in the middle of a bunch of houses, and I think it's just been weeds for several years. In fact, before he purchased it, they actually had to mow like 6-7 foot weeds down. And so I just thought it would be interesting to go take a sample there that hasn't been cropped for at least a couple years. So, and that is kind of interesting, the results that we got there compared to everything else. I would say my ranges were anything from 0 to 350 eggs per 100 cc's of soil, which I have no idea. Is that high or low? 300 sounds like a lot.
Yeah, so that's a coming out of corn, going into beans. We're going to read those as higher than we would have coming out of beans, right? But in Nebraska, for example, our median positive field coming out of beans is around 4,000 eggs per 100 cc of soil.
Whoa!
So that's the median. We see a lot of fields that have very low levels, lower than we're seeing here. We also get fields with 80,000. I don't know, Horacio,
Wow.
What's the highest SCN count you've ever seen?
So from a commercial soybean field, 27,000.
Mm-hmm. So we usually get a couple of those every year.
Yeah, and it's really hard to bring those levels down. And that's one of the, best management strategies to actively sample and identify if you have soybean cyst nematode that you do, and then try to keep those levels low, which is easier than trying to bring higher levels down. And you say 350, correct, Corey?
Yeah, that's my highest.
And that's great, because I consider those low numbers, maybe not even causing much damage yet. But then how cautious we need to be about that, low level, and how in one season it can explode if, we feed that nematode a very susceptible soybean in a very conducive environmental condition. And we have growers that engaged in this experiment that I proposed them. They sample in spring before planting, and they come with very low numbers. And then my request is, let's go to the same site and sample again in fall. And levels of 400 or 200, very low. By the end of that season, we're above that 1,000 levels that Dylan was talking about.
So maybe we should do a follow-up following these soybean fields in the same fields that I did this fall, and see if they went up, or how they were. I would imagine they would go up just growing soybeans there.
Yeah.
That would be excellent, and maybe you guys can have us back. We can do a part 2 after harvest.
Yeah.
Yeah,
So I will say that that 30-acre field that was weeds, that was the one that was 0, had no egg count or anything in it. So I thought that was interesting.
Yeah, and that's actually, it makes sense, right? Even if some of that nematode blew in there over the years from close by fields, it didn't really have anything to really increase numbers. And by nature of soil sampling, we're probing. We might miss the odd random nematode there, but that makes complete sense that that's a new field.
Yeah.
And we actually run into that problem with research. We go out and we try to find some areas that have no nematodes at this point, and we have to go to these new fields or corn on corn fields and work with that. And I'll also say your corn on corn field, with the history of that, was your second lowest one that you had out of that group of fields as well.
Yeah. I would say that our recipe for higher yielding soybeans that we have found is following corn on corn several years. The longer the corn on corn, the better our soybean yields. And I think some of that has to do with just nutrients in the soil, and you got the carbon mixing. But right. I think I've always attributed it to it's probably just less cyst in there.
And that's a big one. I think we've also seen soybean yields not keep up with the progression that corn yields have made in the last several years. But that could actually just be that soybean cyst nematode is building up in these fields across our country, and that's holding those yields back. So, yeah, it's probably a mix of things, but the cysts are actually definitely playing a role.
What's your average soil pH?
Oh, yeah, high sixes, 6.5 to 7. Try to keep it in there.
Okay.
Mm-hmm.
I'm playing around on your profit checker on the website, and I'm going to see, based upon your 350 count field, what percentage you may be losing in profit.
Okay. So does pH actually play a role in soybean cyst nematode?
Yeah, two of the biggest predictors that we've observed are sand content in that soil and soil pH. As those increase, the nematodes do a little bit better. So that can drive yield loss based on those levels.
So the higher the pH, the more cyst?
Yes, correct. For some reason, alkaline soil, higher pH soil, for some reason in those sites, the reproduction rate is higher.
What would you say percentage?
Not a ton.
10%?
Yeah, not a ton of sand. I mean, there's a little bit of sand, but never I would never say I have a sandy field.
Right.
And that's a nice way to target where you're gonna start sampling. Sometimes it's like, where do I start? Should I just take our random cross-section sample? That's fine, but maybe you want to be strategic and have comparisons. What we normally recommend is entryways, areas where soil will move either because of water flowing, high pH content, and then you can have multiple sites where you are evaluating. Using yield maps from previous year, it's another one. Most of my growers have come to realize that they had soybean cyst nematode because we went to those low yielding spots that they have, did everything and still continue yield low, right? So we took a soil sample, soybean cyst nematode.
So I went to the scncoalition.com, ran through the profit checker calculator. I put in Corey's largest number, 350 per 100 cc's. It pre-populated my female index at 45, so I just left it. I put 10% sandy soils and your soil pH of 6.75. I just split your difference for what you told me. I put in a projected yield of 70 bushel to the acre, being pretty conservative for you. And it spit out that his potential loss per $10 bushel soybeans, again, just using round numbers, was over $33 an acre.
I mean, 4.8 yield or loss.
Yep.
Bushel yield loss. So it's $48, right? At $10 soybeans. So in the all years, it's a big deal.
That's percentage, not 4.8 bushels.
Oh, I thought it was
that's
bushel.
A percentage
I'm
of
just
your
looking.
70.
Okay.
Yep. But still, that's a big deal. And you guys said he's got a low count.
Yep.
Yeah.
Is
4%
sand
compared to 30, which with higher numbers? Yes, in that sense, right? And actually, this calculator is becoming very handy to move away from tables that talk about low, high, moderate, right? Because it takes more into account certain aspects of Corey's field.
You say on the sand, that's where they thrive. Is it purely because they like sand, or is it because of drainage? Like, if we just have really well drained non-sandy soil, do they thrive there as well?
So I can take this question. It's a good one because we get a lot of these questions, and one of the comments that we receive is, I farm heavy soil, so I don't have to worry about soybean cyst nematode. And we have a project with the master students where we're purposely going and taking soil texture. So we have a range of different texture, sandy to very heavy soils and everything in between. The sandy soil allows the animal to move faster, and it does move very short distance, right? It also creates more of additional abiotic interaction. So those soils will get dry faster, plants will stress faster, and you put that pathogen there and it causes more significant yield reduction. Once the nematode hit the root, doesn't matter if it's in a sandy soil or in a heavy soil, it will reproduce. So we may be delaying, that dramatic yield reduction that we see in sandy soils when we plant in heavier soils with nematode. But when that population builds up and it's pretty much across, then we're in pretty much the same fate.
Okay.
Yeah.
So what's the future look like? What are you guys excited about? Got anything coming down the pipeline? Major breakthroughs with this?
Well, I think in the short term future, I think the most exciting thing is peaking the resistance source is making it into really good genetic backgrounds. It's been around for a while. When 88788 started to fail, peaking showed up on the scene. But for breeding, it takes a good decade to get good changes. And now we're getting that resistance source in really good backgrounds. And we've just seen it doing really well in variety trials. That's the short-term excitement. But even with that, PI 88788 is still effective, and we need to keep rotating those, if nothing else, but to save peaking from breaking in the same way that 88788 did. There's potential traits coming out for soybean cyst nematode in the future. But again, these things take time, so we might not see any of that for who knows how long. But that could really change the system here. Maybe even in the longer term, breeding is still going on and we're identifying new resistance sources out there. But it takes so long to get those into a variety from a pipeline. But we'll see that in the long term. Nematicides or nematode protectant seed treatments, there's going to continue to be new stuff available. I hope those are always better than the previous generation. So I think that's going to continue to be a stopgap. But really the big thing now is just getting that awareness out there so we can maintain the resistance genes we have for as long as possible. This is the same problem in different fields. We've got limited tools to control these pests, and we need to use them carefully so that they'll last as long as possible. Unlike herbicide-resistant weeds or other problems, you can't blame this one on your neighbors. This one, everyone's got their own nematode population, and they get to choose to manage it how they want. So think about that on your own farms and make those genes last as long as possible. Same thing with all these tools. Use them properly so they last for you.
So I've got the Iowa State University suggestion, right, on how to kind of combine everything that we talked about today. I didn't want to go the whole episode without talking about our cyclones. But ultimately they did. They talked just like you said, Dylan, about how to kind of go about it. They suggest first year plant a PI 887088 resistant soybean, second year a non-host crop such as corn or wheat or oats, alfalfa, then switching up in that third year to a different resistant soybean, whether it's peaking or new technology that comes out. Go back in year 4 to a non-host crop, and then potentially in year 5 a possibility of a SCN susceptible soybean. So I don't know if that's where you would rely maybe on a treatment, maybe on pairing that type of a suggested seed treatment with some natural microbes that might be able to combat that issue, followed by a non-host corn crop in year 6 as a potential 6-year rotation. Mm-hmm. I know when I said that, both of them, their eyes got really big on that year 5 suggestion.
What I'll say is you'd be hard-pressed to find a susceptible soybean because everything's got 88788 and a few things have peaking. So we've still got a lot of 88788 in the germplasm. in certain maturity groups, you will find susceptibles still. So that would make sense. But where you guys are, I don't know if there's any susceptibles out there. But definitely rotate between those two sources. And every now and then you might see a different source. Try different things. Keep those nematodes confused, basically, so they don't learn how to overcome it.
Yeah, when I look at a whole seed portfolio and say there's 30, under this one company from real short to up to 5.0 maturity, you might find one or two. It's almost about the same number that you find just Roundup Ready now.
Yeah.
It's getting down there.
Yeah.
So I've
from,
got one
from our
more.
conversation.
Go ahead.
Yeah. No, from our conversation, I will caution planting a susceptible, right, in any instance, just because of how fast the nematode can reproduce. And the SCN Coalition, we have a very nice infographic of how explosive these populations can be when we provide a very susceptible cultivar. Now, like Dylan mentioned, most of our PI 8878 by definition is already susceptible at some degree to the populations of nematodes we have. But I think for the audience, there's a really easy way to remember a nice integrated management strategy, the triple R management to SCN. Rotate, rotate, and rotate. Rotate to a non-host, rotate different soybean varieties. Don't feed this animal the same generic material because it will adapt, and rotate sources of resistance.
Right.
And on top of that, sample your field so you know if you have it, so you can monitor those numbers and consider a seed protectant, because you're going to have other stuff other than soybean cyst nematode also threatening your soybean yield.
He did such a great job of wrapping it up. I could just be a jerk to you and say, that's it. We're done.
This is important.
But I know you got one more question.
This is important. This is out in the weeds. Everything that we talk about in ag, it seems like it's just a Band-Aid trying to get to the next technology. It's, we're just trying to kick it down the road. So hopefully in 8 years, we know that this gene is coming or this trait or whatever. What about dealing with the actual source? You said these are an animal. We've been able to breed animals for years. Can we harvest these things and manipulate them and maybe make like a sterile male or something like that to release back and actually just get rid of these things?
Anything's possible. So I think you're probably talking about what they might do with managing mosquito populations, right?
Right.
Yeah, that's definitely something we've all talked about. And it definitely could be neat in the future. It's tough, though, because it's hard to get them all. And even if some survive, you could knock those populations down, but they grow so fast. I've seen fields go from 1,000 eggs to 25,000 in 1 year. So you really got to think about managing this at any level. And if we can't get them all, it's going to keep coming back and getting us.
Yeah. I'm glad that you said that, because I had wrote it down earlier, but I wrote it down as stale mail.
Stale mail.
And that's just one that needs a shower.
I was thinking of didn't we do that with like the bot fly or something in cattle that we'd actually, we've gotten rid of a lot of really bad things.
Yeah. No, that's a way to think about science and next research, Corey. I totally agree with you. And one thing that we were talking about, how excited we are of new products coming. One is Nemasphere from BASF, for example. It's a transgenic soybean. And what it does, it expresses this cry protein, like the BT that we know from exactly the same, but it's one that targets this nematode. So, you just mentioned it. How can we do the same thing? And it's already out there. It's going to be hopefully available in 2028, pending registration and everything, right? But can we do something else? Can we modify it in a way that, those feeding sites now are producing only males or completely shutting down? So
and just create a lazy male.
Yeah. That's right. That's a great comment.
Well, I only had one more question left.
Oh, you got a question? Okay.
Is that okay?
I thought you were going to wrap it up.
I have never seen a cup like what you've been drinking out of. There you go.
Oh,
What
so
is
this is yerba mate. This is like a tea. It's a South American tea, very traditional in Argentina, Paraguay, Uruguay, parts of Brazil. You put hot water. There's a filter straw here, and you just drink the tea, and it's highly caffeinated. You know,
the cup looks like it's got feet.
which is a gourd. It's like a little pumpkin that you cut, and then you put little things so it doesn't triple
the
over your
top?
table, and then
That's a filter.
Oh, that's the filter.
Yeah.
The top is the filter.
yeah. No, this is actually the tea. This is the tea here that you put,
Oh.
and then the straw has a filter, so it doesn't get all the tea inside.
I was gonna have to get Corey one of those for his bourbon.
That's very fancy.
Filter your bourbon.
Yerba mate. You should check it out. Yerba mate. I mean, Madonna drinks it. Come on.
That's not the celebrity name I would have expected to be shouted out on a Farm for Profit episode, but we got it.
Wrote it down.
Well, gentlemen, this has been a pleasure. I like geeking out on topics that I don't know a lot about, and I appreciate the way that you were able to describe it to our listeners. I also appreciate the SCN Coalition for helping put this together and rounding you two up as far as informing our audience. I think you provided a great value, and I hope this is a resource that can be shared and passed along to kind of spread the word.
I think we better connect this fall when I take my samples on these fields and see how we did.
That'll be great.
Awesome. Well, gentlemen, I want to thank you again, and listeners, I appreciate you hanging in for this episode. Hopefully it brought you a lot of value. So until next time, have a good one.